Hi, I'm Michelle Jacob. I'm a professor in the Department of Neuroscience at Tufts University School of Medicine in Boston. And today we're going to be welcoming you into our lab and telling you about our research project on CTNNB1 syndrome. We have been working on this project for a couple of years, and what I thought I would do is start in my office and show you a couple of data slides that give you the big picture view of the work we're doing, and then take you into the lab and introduce you to the members of the lab that do the work. So we'll start with a couple of slides. So we were contacted several years ago by one of the moms of a child with CTNNB1 syndrome. And that really started everything for us. That led to our introduction to ACT and to Lauren and Nicole and their families. And that motivated my lab to start to work on CTNNB1 syndrome. We have been studying that gene for many years, looking at gain of function, overexpression of beta-catenin, loss of function, loss of beta-catenin expression in the brain in mouse models. And when we learned about CTNNB1 syndrome, we decided that we would make models that were more representative of the disorder, which basically meant creating a mouse that had germline loss of one copy of beta-catenin to be haploinsufficient like the children. And when we created this mouse model, we were supported by ACT as well as by an NIH grant that we wrote. And we're happy to say that the mouse model looks very promising. And you need models to learn more about the disorder and also to find treatments. And so that was our real goal, was to create an in vivo model where we could test drug treatments and make sure they're effective and safe. And we are also expanding that to human cell models. And I'll tell you about that a little later on. So when we created the mouse, it's a global heterozygote for beta-catenin, or CTNNB1, the gene that encodes the protein beta-catenin. We do see decreases in beta-catenin at the mRNA level, meaning less translation, less transcription, and less translation because there's less protein. So we looked at different parts of the brain, and we've looked at skeletal muscle. And the mouse shows phenotypes similar to the children. The mouse does show learning disabilities in behavior tests that we've done for learning, motor learning, associative learning, and reduced muscle strength. So we have been aware of native proteins expressed in cells. Beta-catenin is expressed in all cells of the body, and there are other proteins that regulate its levels. It has to be very tightly regulated. Levels too high or too low are associated with negative effects on learning and memory. So we wanted to test drugs that manipulate these regulators. In particular, we wanted to test a drug that was an inhibitor of a negative regulator. And so we were really happy when we did this experiment. We got great results. So this is, so far, one of our most promising treatment strategies, where we took young adult mice, 6 weeks old, and we treated them for 5 days, every day. And we found incredible increases in the learning ability of the mouse. When the mouse was treated with vehicle, the heterozygote mouse, it did not do as well in learning as the wild type you see here. But when we drug treated the heterozygote mouse, it came up to wild type levels. And you can see that in this histogram here. So this looked like it gives significant improvements in learning. It also gave us significant improvements in muscle grip strength. So the heterozygote mouse is less able to show grip strength compared to wild type littermates, but with the drug treatment, it significantly improved. And so this looked very promising. We looked at beta-catenin levels, and they come back up to the normal control littermate levels. We don't want the levels to be too high or too low. So this was a really important aspect of the work with the drug treatment. We want to make sure there are no adverse effects. We treated wild-type mice with the same drug treatment protocol, and so absolutely no adverse effects on behavior, on learning, on muscle grip strength, and on beta-catenin protein levels. So this preliminary data is something that's really driving us right now because it's the first evidence for a potential treatment for CTNNB1 syndrome. We have gone on and tested one other drug that didn't look as promising. We have a third one that looks promising for increasing beta-catenin levels, and we're actually doing the behavior test this coming week. And so we'll know if that one looks good. And we're looking at another possibility. We're considering a drug that's been produced by a pharmaceutical company that may be effective, and it's early days, we haven't tested it yet, but we're talking to the company about this. So that would be very exciting. We want to know more about the underpinning of the disorder, what are the molecular changes that cause the symptoms, and that's work that's ongoing in the lab, and you'll meet some of the scientists in the lab that are doing this work. And we also want to look for progressive changes over time so we can really understand the disorder and its sequence. But I think a really important aspect that we're getting geared up to do right now is to go from the in vivo mouse models to in vitro human cells. So drugs that work in mouse have to also work in the human to be something that could ultimately go to a clinical trial. And that's what we want to test. So we're creating human-derived cells. You start with like a blood cell or a skin cell, you create a pluripotent stem cell, and then you can differentiate that to a neuron, typical of what you see in the brain or the spinal cord, differentiate it to skeletal muscle. So we're using two approaches. We're going to start with kind of like a gold standard. It's a cell line from a human was normal, skin cell fibroblast, that's been differentiated and deeply characterized to have absolutely no pathogenic mutations. And we're getting a CTNNB1 heterozygote of that cell line made for us now. And as I said, we'll differentiate it to neurons and muscle. But we also want to look at the mutations in the patient. So this will be a complete loss of function of one copy of beta-catenin gene. The patients have mutations all along the gene, and we don't know if those have different effects depending on whether or not there's a product made or whether or not they will all show a loss of function. It's something that we feel is really important to test and to really understand whether or not the beta-catenin protein is completely missing or if there's a fragment that either retains function or creates what we call a dominant negative, blocks the function of the one wild-type copy. So for that, Wendy Chung, who is an MD PhD that I think is familiar to most of you, Wendy Chung and I have looked through all of the data that's in the Simon Searchlight. This is the particular mutations of different families that contributed information, the phenotypes of the children. We went through all of it, and we've picked out 5 different mutations across the entire gene. And Wendy is having pluripotent stem cells derived from those patient blood cells. And we'll be working with those to really understand beta-catenin protein expression levels. Is there a product made? What are the molecular changes in relevant human cells, disease-relevant human cells? And importantly, the drug treatments that work in the mouse, we now plan to test as well in the human-derived cells. So we're really excited about this work, and I think it's going to be very informative for moving things forward to ultimately what we hope will be clinical trials. So at this point, I'd like to take you into the lab and introduce you to the people who are actually doing the work. Now I'll be introducing you to 2 other members of the lab. Dila is a visiting scientist from Germany who's looking at a related project and changes in synaptic spine density in a mouse model. John Alexander, who is recently promoted from a senior postdoc in the lab to a research assistant professor, so we're very excited for him. And John has done the bulk of the work that I've been showing you, and he's going to give you a little description now of the types of behavioral analyses he does with the heterozygote, CTNNB1 heterozygote mouse model.
Hello everybody. So we use a variety of behavioral tests to not only assess cognition in the mice, but also motor learning and motor capabilities and strength. So I'm just going to sort of talk you through some of our typical behavioral assays that we do in order to assess these things. So one of the things we do that's great for spatial learning is what's called the Barnes maze. Basically, the mouse is placed on a large platform with 40 holes. Underneath one of the holes, as you can see down here, is a little escape hatch. Now, the mouse basically is trained to learn where that particular hole is because they don't like being out in the light. So a mouse with normal learning, after a few training trials, will go basically directly, once it's placed on the platform, into the hole. Now, Mice with cognitive difficulties often don't learn nearly as well and still take additional time or less direct routes to actually get to the escape hole. So some of the studies we've done in our previous mice have been on the Barnes maze, and as you can see on the sort of the day one of training, the blue line and the red line represent the control versus the mutant mice, and you can see both of them start off the same. However, by the day 3 of training, the blue mouse goes pretty much directly to the goal hole, whereas the mutant mouse is basically still traveling around a lot until it finally does work its way back to the goal hole. And we can basically test this at probe trials at different time points to assess then how well they've maintained the memory of the location of those holes. And we get all sorts of things like the latency, which is obviously our main measure in this assay, all the way to the number of errors they create during each particular training trial. We have software in the behavior core that allows us to not only trace the images like you saw before, but also create relative heat maps that allow us to understand where the mouse is spending most of its time. In this test is called the three-chamber test, and it's a way to assess sort of not only social behavior but also social learning in mice. So during the bad habituation phase, you can see the control mouse spends pretty much equal amounts of time within the 2 chambers, which are both empty at this point. However, when you introduce a mouse into one of the chambers, then your test mouse spends most of its time interacting with the control mouse. But to assess memory or to assess social learning, then what we do is put a novel mouse onto the other side compared to this old mouse here on this side. And a normal mouse will actually then switch and say, hey, this person's new, I want to go spend time with them. And you can see in different mutant mice that we study that some of the mice have phenotypes in the social behavior versus non-social, as well as in the social learning. So again, this is another assay that we use to assess learning in these mice. To assess motor phenotypes, we use multiple things, including grip strength test and the rotor rod, in which we place a mouse on a spinning rotor and basically measure the latency until the mouse falls off. And this gives us an idea of motor coordination and learning. And these kind of tests basically allow us to assess how well these mice are able to not only move coordinated at slow speeds, but then adapt to increasing rotation rates in order to basically sort of test muscle learning.
And now we're going to welcome you into the lab. So this is the space that we work in. This is an undergraduate student, Gina, who's working with us for the summer. This is the area where we do all the molecular biology type work to analyze changes in the tissues that we're interested in, the brain and skeletal muscle. An example of a Western blot looking for molecular changes in the heterozygote mice versus the wild type control mice. So this is Leanne Vasquez Ramirez, who's a member of the lab, and she'll be telling you a little bit about herself and the great success she's had with presenting the data from the lab.
Hi everyone. As Dr. Jacob mentioned, my name is Leanne Vasquez. I have been part of the Jacob Lab for about 2 years now, and this is the project that I was working on remotely during my time as part of the Jacob Lab. And so I got to create and design this poster with the data that was collected during that time, I was able to present it at the Summer Program Symposium, in which I won the first place award. And I also got to present this poster at the Annual Biomedical Conference for Minority Students, in which I won an award for the neuroscience category.
Thank you. We hope you enjoyed the tour and getting a sense of the work we do in the lab and the people in the lab.